Okay, let's go ahead and finish up talking about respiration. Let's start off talking about aerobic respiration. So the rest of aerobic respiration, now again, keep in mind the first part we had two things going on. We had the catabolism of glucose and glycolysis. So glucose is catabolized or broken down, and you end up producing two pyruvates. It also loses some electrons. The electrons that are lost end up yielding reduced NADs. And finally, there's some anabolism in this process. You take a total of 4 ADP molecules and add phosphorus to them to make ATP. So there's both anabolism and catabolism, and you'll notice how they're linked together. So let's go on and talk about the rest of the catabolism of glucose first. So we're going to start and take that pyruvate and break it down so that all we have left is carbon dioxide molecules, and then we're going to start making lots and lots of ATP. So the first step, or the next step that occurs, is I will call it the intermediate step, but in truth it's actually the oxidation of pyruvate. So we start off with pyruvate, and you'll see the 3-carbon molecule here is pyruvate. We oxidize it, which means we're going to donate electrons. In that process, we're going to break off a carbon, make our first carbon dioxide, and then finally we'll produce a 2-carbon molecule called acetyl. Now that acetyl binds to something called coenzyme A, but later on the coenzyme A will break off, and so you can see that there's a net yield of, you're starting off with a 3-carbon molecule and you're producing carbon dioxide and a 2-carbon acetyl group. Okay, that acetyl-CoA is carried inside the mitochondria, and that's where the citric acid cycle is going to occur. Citric acid cycle is just that. It's a cycle where a 2-carbon molecule is going to enter the matrix and join with a 4-carbon molecule to form a citric acid. Again, seems a little bit counterintuitive. We just broke this thing down to make a 2-carbon molecule, but this is just a temporary step, and we're going to break those 2 carbons apart and end up just recycling that oxaloacetate. So you take the 2-carbon molecule, join it to a 4-carbon molecule, you make citric acid, and the cycle starts with citric acid. The citric acid is going to be broken down slowly, and 2 carbon dioxides will be broken off, and then we're also going to produce some reduced NAD and then another reducing intermediary, something called reduced FAD, which is very similar to reduced NAD. Again, keep in mind this cycle occurs twice for every glucose that goes into glycolysis, because we have 2 pyruvates for every glucose that goes into glycolysis. So here's sort of a nice picture of our 2-carbon acetyl-CoA, and here it is here, joining a 4-carbon oxaloacetate to form citrate, or citric acid. From the very beginning, we're going to oxidize that citric acid into the electrons that are released are going to be used to make a reduced NAD. A carbon dioxide is broken off, another carbon dioxide is broken off, and we're going to do some more oxidizing. We're going to use some energy to make ATP, and then this 4-carbon molecule is going to break off a few more electrons until it's finally reverted back to the oxaloacetate that can be recycled and used again in the next step of the citric acid cycle. So again, it's 2 carbons plus 4 carbons makes 6. You break off a couple of carbon dioxides, you reduce a whole lot of NADs and an FAD, you also make an ATP along the way. At that point, okay, of the glucose that we started off with, we've taken the glucose and we've broke it into a 3-carbon molecule. That 3-carbon molecule lost 1 carbon in the first step of the oxidation of pyruvate, and then another 2 carbon dioxides were produced in the citric acid cycle. So it's fair to say that we have completely catabolized, completely broken down that glucose to make CO2. So the glucose is all gone, and we need to think about what's left. Well, in the process, we've used the energy from those glucoses to make a total of 4 ATPs so far, and then we've also made a lot of these reduced NADs and also some reduced FADs. Okay, so what's left then is these guys here, these reduced molecules, have the ability to be cached in something called the electron transport chain, and we can use those to make ATP. Okay, Again, what we're going to do is these electrons have been reduced. We need to now oxidize them. We need to donate their electrons somewhere and use that the electrons to phosphorylate or to make some more ATPs. And that's what the electron transport chain is. The electron transport chain, what you do is you donate electrons to molecules that are found in the inner mitochondrial matrix. Those molecules are sometimes called cytochromes. There is a complex of electron carriers, and when you donate those electrons, the electrons can be used, the energy from the electrons can be used to pump hydrogen from the inner mitochondrial matrix into the intramembrane space in between the inner and outer mitochondrial membranes. So there's a space in between the outer mitochondrial membrane and the inner mitochondrial membrane, and we pump hydrogen there using the electrons, using the energy from the electrons that we've been collecting on our NADs and FADs. What happens is we build up a gradient. There's way more hydrogen ion on the outside of this membrane than there is on the inside, and that also builds a charge gradient. So we call this a chemiosmotic gradient. There's both a more positive charge here, so there's a chemical charge pulling this hydrogen more towards the inner space, as well as there being a concentration gradient. Hydrogen can't cross across the phospholipid bilayer. It can cross through a special enzyme called ATP synthase. Okay? This ATP synthase is a special enzyme that can allow the hydrogen to pass through and use the energy of it passing through to phosphorylate an ATP. So two steps go on. First we make the gradient by using the electrons, and then once the gradient is made, we allow the hydrogens to move back in, and those hydrogens that move back in, the energy of them moving in can be used to make ATP. Okay, this leftover hydrogen, these leftover electrons, and oxygen are finally used to make water. Okay, again, electron transport is a step where the electrons, as they lose energy, as they slowly go from higher to lower energy levels, they're able to pump hydrogen, and then hydrogen makes a gradient. And as the hydrogen moves back across the gradient or across the membrane through the enzyme ATP synthase, the energy that's released is used to make ATP. Okay. Again, sort of a summary slide talking about the electron transport chain. The electrons being used to pump hydrogens into the intramembrane space, and ATP synthase using the passing through of those hydrogen ions back into the inner mitochondrial space to make ATP. Again, the extra electrons, oxygen, and hydrogen are used to make water. Okay, so here's a neat summary of the overall process of cellular respiration. You make 2 ATPs net in glycolysis, you make 2 ATPs net in citric acid cycle, and you make a good number more, about 32 or more ATPs in this process of oxidative phosphorylation that occurs in the electron transport chain. Now again, that's it. That's all there is to cellular respiration. Again, it seems like quite a lot, but I think if you review it, you shouldn't have too much of a problem. The thing you need to do is break this down into the 4 steps. The 4 steps are glycolysis, the intermediate step, or the oxidation of pyruvate, the citric acid cycle, and then the electron transport or oxidative phosphorylation. For each step, look at what you start with, what you end with, how many ATPs were made, where does it occur. Again, follow along with the reduced NADs because they're the ones that carry the energy when we go through aerobic respiration. So I put in a few extra review slides, just a quick review for glycolysis. Make sure you know that a glycolysis occurs in the cytoplasm. It's anaerobic, which means it doesn't require oxygen. You put glucose and 2 ATPs in, you end up with 2 pyruvates, which are 3 carbon molecules, 2 reduced NADs, and a total of 4 ATP are produced, but the net gain from this is 2 ATPs, because again, remember we had to input 2 ATPs to start off with. The next thing we can think about is fermentation. Again, in the absence of oxygen, the cell has to recycle the reduced NAD back to the oxidized version, and when that occurs, we call it fermentation. When fermentation occurs in animal muscles, we produce lactic acid or lactate. When it occurs with yeast, ethyl alcohol or ethanol is produced, and there's other products, potential products produced with bacteria. Okay, the next step is oxidation of pyruvate. Pyruvate was a 3-carbon molecule. When we oxidize it, we end up producing a 2-carbon molecule. That 2-carbon molecule, the acetyl, joins with the coenzyme A and will go on to the citric acid cycle. This oxidation yields 1 reduced NAD. The citric acid cycle. We start with a 2-carbon acetyl group, join it to a 4-carbon oxaloacetate to form a 6-carbon citric acid, and then 2 carbon dioxides are broken off. A 4-carbon oxaloacetate is going to end up being produced. We also produce an ATP and many reduced NAD and FADs for every time we go through the citric acid cycle. Finally, we can talk about the electron transport chain. The electron transport chain starts at the intermitochondrial matrix, where reduced NAD donates its electrons to the cytochromes, or the electron carriers that are found on the cristae, or the intermitochondrial membrane. The cytochromes actually will use the energy from those electrons to pump hydrogen to the outer mitochondrial space, forming a gradient. And finally, ATP synthase, a special enzyme that's found on the inner mitochondrial membrane, will allow hydrogen to cross and use the energy to make ATP. Again, the products are ATP and water for the electron transport chain. Okay, with that I'll stop. Make sure you spend some extra time, think about things other than glucose that can go through cellular respiration, and spend some time with this. Again, if you review it several times, you may find that it's not quite as awful as it seems at the first